So in this video, we are going to study a few topics of genetic engineering and biotechnology. So the first thing that we are about to study is the PCR, which is nothing but the polymerase chain reaction. And the purpose of this particular process is to create large quantities of a specific target sequence of the DNA. So prepare multiple copies of a particular DNA sequence. So it is useful when only a small amount of DNA is available for testing and you need more. For example, in case of a crime scene, you need more samples of blood, tissue, hair, etc. You have a small sample and you need more of DNA. So PCR, it occurs in a particular object known as a thermal cycler, and it has three processes. It's a repeat procedure of 3 steps, and one cycle of the PCR will give you 2 raised to the power 1 copies of DNA. 2 cycles will give you 2 raised to power 2 copies of the DNA. So basically, n cycles will give you 2 raised to power n copies of the DNA. It's not just double every time, it is 2 raised to the power of the number of circles. So the 3 steps that occur in the thermal cycler are denaturation. Denaturation refers to the process wherein the DNA sample is heated. And it is separated into the 2 strands. So DNA sample is heated to separate into the 2 strands. The second step that happens after denaturation is annealing. So annealing refers to the process in which DNA primers, they attach to opposite ends of the target sequence. So I'll explain this process diagrammatically as well. And the third and the final step is elongation. In this, a heat tolerant, because remember the DNA is right now in a very hot state, a heat tolerant DNA polymerase TAQ, that is one example. It helps to copy the strands. So that is what happens in a polymerase chain reaction. So let's see what the diagram is. So basically, if you have this DNA segment, it's going to unwind just like it happens in DNA replication. And these 2 segments will be cut by using the DNA primer, so they act as scissors. And this is now going to be copied into many strands. You can get multiple copies of the strands depending on the number of times you run the reaction. So that is polymerase chain reaction. The next process that you need to know, what happens after polymerase reaction, is another thing that can happen, which is gel electrophoresis. Now, these all concepts are related to one another, that's why I've put them into one single video. So in gel electrophoresis, it's a technique that separates the DNA fragments according to their size. So let me draw a diagram of the components. So this is a tub-like structure or a plate-like structure. So samples of the fragmented DNA will be placed in this. It is a well made up of agarose gel, and there are 2 electrical circuits that are put over here. One is the negative side, negatively charged cathode, and the other side is a positively charged anode. And obviously the agarose gel would be the conductor. Now gel is placed in a buffering solution and the electric current will be passed through the gel. Now the movement of fragments, in which direction do you think it will take place? Now DNA, as a molecule, it has negative charge because of the phosphate group. So if it is negatively charged, it will be attracted towards the anode. That means movement of fragments takes place in this direction. Now the smaller fragments, they travel faster because of their small size. They will get impeded less by the gel matrix and they will travel faster. The bigger fragments will take longer to move towards the positive anode. So as a result, after some time, the fragments are separated according to their size. And now the size can be calculated in kilobases by comparing those sizes against a known industry standard. So after that, a photograph of that entire gel is taken and it gives you a picture like this. So the big fragments would be at one end, and then you have small fragments depending on the distance they travel. And this kind of a photograph that you get is known as a DNA profile, which is in fact the next thing that we are about to study. So what is this DNA profiling? It's also used in a lot of forensic investigations, and it's a technique by which individuals can be identified on the basis of their respective DNA profiles. So you can take profiles of different people and it's going to be unique for every person. So how this is taken, now there is a special concept that you need to understand for that. Within the non-coding region of a person's DNA, within the non-coding part of the DNA or the genome, there is a specific DNA known as a satellite DNA. What is the satellite DNA? This is basically composed of long stretches of DNA that are made up of repeating segments or repeating elements of bases called short tandem repeats. So these repeating elements, these are known as short tandem repeats. And these repeating elements, they form long stretches of DNA that are present inside the satellite DNA. Now these repeating sequences, they can be cut to form fragments. So these repeating elements, they are cut, they form fragments. And the enzymes that are used to cut them are known as restriction endonucleases. Restriction endonucleases. So these cut DNA at specific sites, and as individuals have a different number of repeats in a given sequence, they will all generate unique fragment profiles. And you can compare these profiles with one another to find a correct match. And how these profiles can be compared? Once the fragments are there, you can make them undergo the process of gel electrophoresis. And from gel electrophoresis, you can get these different kinds of plates. And from these plates or photographs, you can compare the person. For example, if you found this kind of a profile at a murder scene, and you have two suspects, one is this, and the other is this. So you can easily see the first one and the second one are a match and the middle one is not. So that is how the fragments can be compared. Now there are two applications of DNA profiling. One is to determine paternity. So for that you need profile of the mother, the child and the father. And second is forensic investigations. Murders or wherever part of the tissue or hair samples they are found. So that is all that you need to know about DNA profiles at this stage.